Good afternoon. I hope you all enjoyed the lovely lunch we had. So, just a reminder, there will be a brief Q&A session following each presentation. We kindly ask you to hold onto any questions that you may have for the presenter until the Q&A session. And without further ado, Trey, please take it over.
Hi, everyone. I'm Trey Moore. I'm a senior in the College of Arts and Sciences studying neuroscience, and today I'm going to be presenting my project titled Controlling Brainwaves to Treat Alzheimer's Disease. Now, as you may know, Alzheimer's disease is the most common neurodegenerative disorder currently affecting over 6 million people in the United States alone. Despite extensive research into potential therapies, disease-modifying treatments have yet to be elucidated. However, we do know that the main biological culprit to cause Alzheimer's disease is aggregation of the amyloid beta protein. The amyloid beta protein is this really sticky protein that, when produced, floats throughout the brain and clumps together with other amyloid beta protein molecules, and they form these toxic plaques. And these plaques are what's thought to cause a cascade of other pathologies to follow that include neuroinflammation, cell death, and then lead to dementia and memory loss, as most commonly associated with Alzheimer's disease. Now, there's a few ways that this amyloid beta protein can be cleared from the brain, but today I want to focus on microglial phagocytosis. Now, microglia are the brain's resident immune cells, and they're responsible for a variety of different processes, which can be both good and bad in Alzheimer's disease. Now, as you see up here, microglia can be really bad in the Alzheimer's disease brain through neuroinflammation and excessive synaptic pruning. However, it can also be really beneficial in the Alzheimer's disease brain, as it's been shown to be able to phagocytose or eat up these amyloid beta plaques and clear it from the brain. So getting these microglia to do this job can be really helpful in clearing amyloid beta. But in Alzheimer's disease, microglia aren't able to do this. They're just overtaken by the amyloid beta protein. So our proposed mechanism of being able to recapture this ability for microglia to clear amyloid beta from the brain is through neural oscillations. Now, you might have heard of oscillations be referred to as brain waves. These brain waves can be recorded using EEGs or other invasive monitoring techniques. And what you see on the top left up here is what one of these signals might look like. Now, this signal can actually be parsed out into different frequency bands, such as delta, theta, and gamma. In this project specifically, we were interested in gamma waves, as they've been associated with aspects of cognition, such as learning, memory, and attention. Interestingly, these gamma waves have been shown to be notably aberrant in Alzheimer's disease patients when compared to healthy individuals, which you can see an example of on the right. When you analyze these oscillations, it's important to remember that power is amplitude. And when we say that we're evoking gamma waves in Alzheimer's disease brains, it should be associated with an increase in the amplitude of the wave. There's a method that does just this, and it's called gamma entrainment using sensory stimuli, otherwise known as GENUS. Now, this was first introduced in the 80s, but it wasn't until a few years ago that this method was used to show benefit in the Alzheimer's disease brain. A group at MIT was able to use this method and show reductions in the amyloid beta protein that I mentioned in a mouse model, as well as improvements in cognition and reduction of neurodegeneration. This was really exciting, and it moved to studies in human patients, but the same effects on the amyloid beta protein wasn't shown in human patients. Now, interestingly, there's another way to evoke these gamma waves throughout the brain, and it's through manipulating a specific population of neurons, of brain cells, called basal forebrain parvalbumin neurons. This is called the BFPV. And the BFPV neurons project all throughout the brain and have targets throughout cortex, the hippocampus, and the thalamus. They really just project all over the brain. And there's been a few studies that look at the role of stimulating these neurons and generating these gamma waves. And you can see on the bottom that when these neurons are activated at different hertz, gamma can be recorded throughout the brain. Specifically, when they're activated at 40 hertz, you can see gammas generated throughout the brain. So we were curious if generating gamma in this way could provide similar therapeutic benefit as demonstrated in the GENUS studies. So to do that, we performed surgery on 5xFAD transgenic mouse models of Alzheimer's disease. This means that mice were genetically modified to be able to exhibit the amyloid beta protein demonstrated in human AD. We then performed surgery on these mice, and we injected this virus in the BFPV on each side of the brain that makes these cells responsive to light. We then implanted these optic fibers just above the injection site and implanted two recording electrodes, one in the medial prefrontal cortex in the front of the brain and one in the dorsal hippocampus that's in the back of the brain to measure these gamma waves. We then let mice wait for 6 weeks for the virus to be expressed and then injected this compound called methoxyxo4. Now, this is just a compound that binds to the amyloid beta protein in the brain and makes it fluoresce, so we're able to track it later on. We then treated mice for 1 hour per day with this laser stimulation that you can see on the bottom right. Is when we shine this blue light on the cells that we injected this virus into, it'll drive the cells to fire. And we can do this and deliver this treatment at 40 hertz and cause these cells to fire 40 times a second. We did this for 1 day, or 1 hour per day for 1 week, and then collected the tissue and ran it through this fluorescent cell sorting technology so we could determine if the microglia that I had mentioned earlier was able to come back and clear this amyloid beta protein from the brain. But first, we look at the effects of the stimulation on gamma waves by itself. So what you see here are recordings from the medial prefrontal cortex in the front of the brain and the hippocampus in the back of the brain in both control mice and the channel rhodopsin treated mice. On the top, you see there's no real difference from when the laser is being turned on, which is denoted by the blue dotted lines in either the front or the back of the brain. But in the treated mice, we see a significant difference when the laser is being delivered. And you can see that this really kind of confirms that the treatment we're delivering is causing the effect to gamma waves that we had intended to, specifically in the 40 hertz gamma waves, which you can see on the bottom here. And when we did this for each mouse, it just confirmed what we suspected. Gamma waves are being generated in treated mice and not in control mice. So we wanted to see if this difference in gamma wave generation can lead to alterations in amyloid beta uptake by these microglia cells. And we did that using the flow cytometry, which is a cell sorting technology that I mentioned before. And on the left is just an example of how we collected data. The top is the control mouse, and the bottom is the experimental mouse that did receive treatment. And when we did this for each mouse in our study, we see the effects on the right, where conditions on the x-axis, and the percent of total microglia that were labeled as methoxyxo4, meaning that they had taken up amyloid beta, is on the y-axis. And we see that mice that did receive treatment showed significant increases in percent of total microglia that had taken up methoxyxo4 labeled amyloid beta in the brain following our treatment. So there are a lot of future studies to be done. This is just the first step in kind of looking what's going on. And we need to really understand the mechanism that's being activated from this neuronal activation that's causing these microglia to do their job, right? So I think further investigation into BFPV stimulation should look at cerebral blood flow, neuroprotection, different signaling molecules that are being activated from this treatment. And this could potentially lead to certain receptors that might be targeted pharmacologically later on that could be of really cool interest in potentially treating Alzheimer's disease down the road. So there's a ton of people I'd like to thank for this project. First off, I'd like to thank Dr. Rudy Tandy at MGH for allowing me to complete my studies in his lab. I'd also like to thank my advisor, Dr. Felipe Scafrino. He was instrumental in this project, and I could not have completed it without him. I'd also like to thank Kat and Matt, my two best friends at the lab, and they help me every single day. I'd also like to thank Dr. Jerry Lin for treating me in this flow cytometry, the cell sorting technology. I'd like to thank Dr. Pastorino and Dr. Kane for serving on my defense committee. And I'd like to thank Dr. Muscaderi for all of his work as leading the undergraduate neuro program at Boston University. Thank you.
Wonderful work, Trey. Questions? Now it's on. Questions?
I was wondering if you could talk a little more about the application of this into humans, I'm not super experienced with it, but would you need to like put open somebody's head and stick things in it to treat a human? Like, is this realistic or only for developing drugs that work similarly?
Sure. Now, I don't think we're ever going to inject a virus into it and then put lasers into the human brain. It might happen in the future, but I don't think it will. I do think a more realistic application of this is finding a specific receptor on these neurons in the human brain that could be targeted. Now, we can't activate these neurons in the human brain specifically at 40 hertz like we did with the laser. If we targeted it with just a drug, it would just be on, all the time. We'd increase the excitability of these neurons just overall. Now, what we could potentially do is develop a drug that does this and makes them more able to be excited and pair it with the visual genus or the auditory genus that was shown. So we could then entrain 40 hertz gamma in the human brain. That would be really cool, and I think that would be a lot more applicable in the human brain than stimulating it with a laser.
Can you connect what you just said to Alzheimer's in the human brain, like as a treatment?
Yeah, and that's the same thing. We could say we had this hypothetical drug that caused these cells to fire, right? In an Alzheimer's patient, these cells are dysregulated in some way. So these cells are still here, they just can't fire the same way that they would in a healthy individual. So if we had a same drug that could increase the excitability, we could give it to them and then give them this visual or auditory treatment, right? That could then help entrain these gamma waves and maybe clear amyloid beta from the brain. That could be of benefit. So. Thanks so much.
Over.